Hello friends, welcome to SAM Biology class. Friends, today we shall discuss about genome editing. A genome is all the genetic informations of an organism. In most of the organisms, it consists of the nucleotide sequences of DNA. It is only in some viruses the genome consists of the nucleotide sequences of RNA. The nuclear genome includes protein coding genes, non-coding genes, regulatory regions of DNA, and a substantial fraction of junk DNA with no evident functions. In almost all eukaryotes, mitochondrial genomes are also present. In algae and higher plants, photosynthetic organelles known as chloroplasts are present, which also have their small genomes. Genome editing, also called genome engineering, is a group of techniques by which DNA sequences are inserted, deleted, replaced or modified within the genome of a living organism. So, by altering the nucleotide sequence of the genome through editing, we are able to alter the characteristics of an organism. So, this technique helps us to introduce many edits in the nucleotide sequence of the genome. For genome editing, 4 different types of programmable nucleases are used to create double-stranded breaks. These enzymes are meganuclease, zinc finger nuclease, tail nuclease, and CRISPR-Cas9 system. Meganucleases are naturally occurring restriction enzymes. They were first discovered in 1980s. These enzymes are characterized by long cleavage sites which are about 14 to 40 base pairs long. The most widespread and best known meganucleases are members of the LIDADG family of proteins. Second type of nuclease is zinc finger nuclease, which are artificial restriction enzymes generated by fusing a zinc finger DNA binding domain to the cleavage domain of FOC1 restriction endonuclease. Zinc finger nucleases function as dimer. Dimerization is mediated by the FOC1 cleavage domain, which cuts the DNA within a spacer sequence of 12 to 19 base pairs long that flank each zinc finger DNA binding site. Third type of nuclease is TALENs, which stands for transcription activator-like effector nucleases. Like zinc finger nuclease, TALENs are also artificial restriction enzymes generated by fusing a tail DNA binding domain to a FOC1 cleavage domain. These enzymes also function as dimers. And dimerization is mediated by the FOC1 cleavage domain, which cuts the DNA within the spacer sequence. Fourth type of nuclease is CRISPR-Cas9 system, which consists of a single guide RNA and a Cas9 nuclease. The guide RNA contains the essential CRISPR RNA and trans-CRISPR RNA elements. As the recognition of the cleavage site is mediated by the guide RNA alone, CRISPR-Cas9 system has emerged as the most flexible and user-friendly platform for genome editing. All these programmable nucleases consist of 2 different parts. One part is a DNA binding domain which recognizes and binds at a specific sequence of DNA within the genome. The other part is the cleavage domain, which creates a double-stranded break at a specific sequence. This figure shows the basic process of genome editing. In first step, the programmable nuclease recognizes and binds at a specific sequence. After binding, next step is the creation of a double-stranded break. And third step is the repair of the double-stranded break, either by non-homology end joining or by homology directed repair. Non-homology end joining (NHEJ) uses a variety of enzymes to directly join the DNA ends. Non-homology end joining is not accurate, because of which it introduces many errors in the nucleotide sequence of the genome. Homology directed repair is more accurate. It utilizes a DNA template to regenerate the missing sequence at the breakpoint. So by genome editing, we are able to alter the nucleotide sequence of the genome. By doing so, we are also able to alter the characteristics of an organism. So friends, this is all about genome editing. For more updates, please subscribe my YouTube channel, Sam Biology Class.